Hello everyone, welcome to my YouTube channel Pharma Researcher. So this is the video number 18 of Telangana Pharmacist Grade-II exam preparation. The exam which is going to be held on the 30th of November 2024. So this is the series on our YouTube channel Pharma Researcher in which we are covering total of 10 units that are mentioned in the syllabus. So in this series, a total of 8 units have already been covered, and in this particular video, we are going to cover the unit number 9. So whole complete parts of the unit number 9 will be covered in this single video. So in this unit number 9, it is related to biochemistry, which includes carbohydrate, protein, lipids, vitamins, enzymes, and therapeutic areas. So these all topics will be covered in this particular video. And then after this video, we will be left with the unit number 10, which is related to jurisprudence. So that will be covered by today only. So there are a lot of topics. So I'll try to complete the whole video in a single part, or if the question number of question will exceeds, then I'll cover it in two parts. So let's discuss first the PDF related things. So if you need the PDF, so link will be given in the description box. You can download the PDFs, and for the other videos, if you haven't watched those videos, so you can check the playlist on our YouTube channel Pharma Researcher, which is related to Telangana Pharmacist, or all the links have been given in the description box for previous videos, so you can check the other videos as well. So let's start first with the topics. What are different topics that are mentioned in the syllabus? So unit number 9, we have the classification, qualitative test, biological value, deficiency, and diseases related to metabolism of carbohydrates, proteins, and lipids. Then there is a topic of vitamins, which include different chemistry and role of vitamins, enzymes, coenzymes, and minerals as well. And then there are therapeutics, introduction to pathology of blood and urine, lymphocytes, and platelets, their role in health and disease, erythrocytes, abnormal cells, and their significance in abnormal constituents of urine, and their significance in diseases. So these all topics will be covered in this particular video. So let's start first. So our first topic is regarding the carbohydrate, protein, and lipids. So let's start with the first question. So the question is, which class of carbohydrate is considered as non-sugar? So the non-sugar class is polysaccharides. So here option C will be the correct answer. So first we can discuss the complete classification of carbohydrates. So basically there are different classes. There is monosaccharides. Carbohydrates can be classified as disaccharides, then oligosaccharides, and then polysaccharides. So here the monosaccharides are the simple sugars. These are the simple sugars, and they can be categorized as two different classes. First is aldosis, and the other one is ketosis. Means aldosis means in which the main functional group is aldehyde, and in ketosis the main functional group is ketone. So in aldosis there can be different examples like glyceraldehyde, then erythrose, then there can be ribose, then glucose, then glucohaptose. So these are the different aldosis monosaccharides. Then in ketosis there can be dihydroxyacetone, there can be erythrulose, there can be ribulose, there can be fructose, and they can be pseudohaptulose. So these all are the examples of ketosis monosaccharides, and these are the simplest sugars. Then the next class is disaccharides. So disaccharides are those in which there are two monosaccharide units. They are made up of two monosaccharide units, and these disaccharides can be reducing sugars. These can be reducing sugars, or they can be non-reducing sugars. Non-reducing sugars. So these are very important. The examples in reducing sugars and examples in non-reducing sugar, these are very important. So in reducing sugars, the example is maltose. The example is maltose and lactose, while in non-reducing sugar the example is sucrose. So these examples, both reducing and non-reducing sugar example, is very important. Then there is oligosaccharides. Oligosaccharides, in oligosaccharides, they can be made up of 3 to 10 monosaccharide units. They are made up of 3 to 10 monosaccharide units, and the examples can be raffinose. Example can be raffinose, stachyose, stachyose, vervascose, vervascose. So these are the examples of oligosaccharides. Then coming to polysaccharides. Polysaccharide classes are very important. So these are the carbohydrates which are made up of more than 10 monosaccharide units. They are made up of more than 10 monosaccharide units. Now they can be homopolysaccharide, they can be homopolysaccharides, or they can be heteropolysaccharide, or they can be heteropolysaccharides. So in homopolysaccharides, the examples can be starch. Example can be starch, dextrin, starch, dextrin, glycogen. It can be cellulose, it can be cellulose, it can be inulin, dextran, and chitin. While in heteropolysaccharides, the examples can be agar or agarose. They can be gum and gum, pectin. There can be highly uronic acid. This is highly uronic acid is important. Then there can be keratin sulfate, or they can be heparin. So heparin is also important. Sometimes the question can be asked like, heparin is which type of polysaccharide, or which type of carbohydrate. So it is a heteropolysaccharide. So the examples in both homo and heteropolysaccharides are very important. So these are the basic classification of carbohydrates. Now we can move to our questions. So the next question is, which of the following is also known as invert sugar? So invert sugar is sucrose. So here option A will be the correct answer. Next question is, the only carbohydrate which is not having a chiral atom. So the carbohydrate which is not having chiral atom is dihydroxyacetone. So here option C is correct. So basically, what is chiral atom? So chiral atom is a carbon which is having four different groups attached to it. Like there can be four different groups. If one is hydrogen, one can be CH3, other can be C2H5, then it can be NO2, something like that. Means all the four groups attached to carbon atom is different. That is a chiral atom. If it is having similar groups, two similar group, it is CH, then OH, CHO. So if this type of means two groups are different but the other two are same, then it is not a chiral atom. So let's move to next question. So the question is, the glycogen in animals is stored in. So the glycogen which, in animal, it is stored in the liver and spleen. So here option A is correct. Next question is, which of the following is the general formula of carbohydrates? So the general formula of carbohydrate is CH2O whole N, or it can be written as CN H2N ON. For example, for glucose, the formula is C6H12H12O6. So here you can see N is 6 and 2N, that is 6 into 2 12 for hydrogen, and oxygen is 6. So this is a general formula. It can be written as this. So if mentioned as this CnH2 and ON, then that can be the correct answer. Next question is, which of the following is the smallest carbohydrate triose? So the smallest carbohydrate triose is glyceraldehyde. So here option C is correct. Next question is, which of the following is a reducing sugar? So the reducing sugar is dihydroxyacetone and acetic acid both. So here option D, both A and B is correct. Then next question is, which of the following is an example of epimers? So the example of epimers is glucose and galactose. So here option B is correct. So what are epimers? Epimers are those compounds which are similar in molecular formula, but they are different in structure. Basically, they are different in the orientation of a single carbon atom. Okay, on the orientation of single carbon atom, they are different. Now the next question is, Molish test is used for. So Molish test is for mucoproteins. So here option C is correct. Or Molish test is a general test for carbohydrates as well. This is important. Next question is, diabetes is a disorder of which metabolism? So diabetes is the disorder of carbohydrate metabolism. So here option A is correct. Next question is, the most active site of protein synthesis is. So the most active site of protein synthesis is ribosome. So here option B is correct. Next question is, deficiency of glucose 6 phosphate dehydrogenase is the disorder of which pathway? So it is the disorder of HMP shunt pathways, hexose monophosphate shunt pathway. So here option A will be the correct answer. Next question is, which of the following describes the relationship between D-sorbose and D-fructose? So D-sorbose and D-fructose, these are the diastereomers and they are not the epimers. So here option B will be the correct answer. And it is related to superimposable mirror images. So it is B option correct. Next question is, which of the following is the simplest carbohydrate? The simplest carbohydrate is glyceraldehyde. So here option D is correct. Next question is, which enzyme is considered as a principal enzyme for the regulation of glycogenolysis? So for the glycogenolysis, the principal enzyme is glycogen phosphorylase. So here option B is correct. So there are different metabolic processes, different cycles that are going on in our body for the metabolism of carbohydrate, like glycolysis, TCA cycle, glycogenolysis. So these all different cycles, I have added the definitions of all these cycles, so you can read those as well. I have added sufficient content as well for the preparation at the end of the MCQ question, so that you can read in the PDF. Next question is, where glycogenolysis takes place? So the glycogenolysis, it takes place in the cytosol. So here option A will be the correct answer. Next question is, which enzyme catalyzes the reaction which is utilized in both glycogenesis and glycogenolysis? So the enzyme catalyzed reaction is phosphoglucomutase. So this enzyme is involved in glycogenesis as well as glycogenolysis both. Next question is, the condition of destruction of RBCs is known as. So the condition of destruction of RBCs, it is known as hemolysis. So here option A is correct. Next question is, dash is the most common complication of G6PD deficiency. So this we have discussed in pharmacology as well. So in G6PD deficiency patients, many drugs are contraindicated because they cause hemolytic anemia. So this is the most common complication of G6PD deficiency. Next question is, chemically dash occurs as a mixture of amylose and amylopectin. So the mixture of amylose and amylopectin, it is starch. So these are the main chemical constituents as well. We have discussed this in the pharmacognosy also. Then the next question is dash occurs as a highly branched form of amylopectin. So the highly branched form of amylopectin is glycogen. So here option C is the correct answer. Next question is dash forms cell wall of plant cells. So the cell wall of plant cells is made up of cellulose. So here option B is correct. Next question is dash is found in milk. So in milk, lactose is the carbohydrate that is found in milk. So here option A is correct. Next question is dash are simple sugars with 2 to 10 units of monosaccharides. Simple sugars with 2 to 10 units of monosaccharides is oligosaccharides. So here option C is correct. Next question is sucrose hydrolyzed into a molecule of each glucose and so the hydrolysis of sucrose will give glucose and fructose. One molecule of glucose and one molecule of fructose. So here option B is correct. Next question is all members of dash give positive result for Molish test. So the positive test or Molish test is given by the carbohydrate because it is a general test for carbohydrate. So option B is correct. Next question is a sugar on heating with phenylhydrazine, hydrochloride, sodium acetate and acetic acid forms dash crystals of oxazone. So it is the oxazone test which is for carbohydrates only. So in this the heating is with phenylhydrazine hydrochloride, sodium acetate and acetic acid and it will form yellow colored crystals of oxazone. So here option B is correct. It will form needle like crystals. Next question is dash test detects the 3-month average plasma glucose concentration. So this is the HbA1c test which detects the 3-month average plasma glucose concentration in the body. So here option C is correct. Next question is dash is a genetic disorder resulting when the ability to metabolize galactose sugar is hampered. So when the metabolism of galactose sugar is hampered or disturbed, then there can be a genetic disorder, and this genetic disorder is galactosemia. So here option C is correct. Next question is what is the solubility of lipids in water? So the solubility of lipids in water is insoluble. These are the lipids are insoluble in water. So here option C is correct. Next question is which of the following is not a lipid? So fat, oil, protein, vaccine. So proteins are not a lipids. So here option C is correct. Next question is sphingolipidosis leads to improper functioning of. So sphingolipidosis it leads to improper functioning of the brain. So here option C is correct. Next question is atherosclerosis is a hardening of. So atherosclerosis it is a hardening of arterial wall. So here option A is correct. Next question is the melting point of fatty acids depends upon chain length and so the melting point of fatty acid it depends upon the chain length and the degree of unsaturation. So here option D is correct. Then the next question is which of the following is an example of mono unsaturated fatty acids? So mono unsaturated fatty acid means only one double bond is present in the chain. So the example is oleic acid. So here option A will be the correct answer. Next question is the high content of triglycerides is seen in. So the high content of triglycerides it is seen in the chylomicrons. So here option D is correct. Next question is sphingolipidosis is the disorder of. So sphingolipidosis it is the disorder of phospholipid metabolism. So here option A is correct. Next question is beta oxidation of fatty acids occurs in. So the beta oxidation of fatty acid it occurs in the mitochondria. So here option C will be the correct answer. Next question is rancidity of lipids of lipid rich food stuff is because of. So what is the reason for rancidity? So it is due to the oxidation of fatty acids. So here option D will be the correct answer. This question is also important, rancidity and oxidation. Next question is, which of the following metabolism causes atherosclerosis? So the atherosclerosis is due to cholesterol metabolism. So here option C is correct. Next question is, the degree of unsaturation of lipids can be measured as. So the degree of unsaturation of lipids, it can be measured as iodine value. So here option A is correct. Next question is, dash is a 27 carbon terpene derivative. So the 27 carbon terpene derivative is cholesterol. So here option D is correct. Next question is, dash are a type of barrier molecule made up of long chain alcohol with an ester linked to fatty acid. So these are the type of Bexes are the type of barrier molecules which are made up of long chain alcohol with ester that is linked to a fatty acid. Next question is, cholesterol and ergosterol are the examples of dash lipids. So cholesterol and ergosterols, these are the steroid lipids. So here option B is correct. Next question is, which of the following test is performed for testing the presence of glycerol? So for the presence of glycerol, acrolein test is performed. So here option A will be the correct answer. So tests are also important. One question from the test will be asked. So whatever the test related to carbohydrate, lipids and amino acid or proteins is there. So you have to remember the at least the name of the test you should remember. Next question is, reducing sugars and glycerol shows positive result for dash test. So reducing sugar and glycerol, they will show dichromate test positive. So here option B is correct. Next question is, hypercholesterolemia is a condition of high dash levels in the blood. So hypercholesterolemia, hyper means high and cholesterol means cholesterol, means cholesterol. So here option C is correct. Next question is, formation of plaque inside the arteries is referred to as a state of. So it is a state of atherosclerosis. So here option A will be the correct answer. Next question is, deposition of cholesterol around the eyelids is known as. So around the eyelids, the deposition of cholesterol is xanthelasmus. So here option D is correct. Next question is, deposition of fat on elbows, buttocks, and tendons is known as. So in these area, elbows, buttocks, and tendon area, if the fat is depositing, so that condition is known as xanthomas. So here option B is correct. Next question is, deposition of cholesterol around the corneas is known as. So the cholesterol deposition around cornea, it is known as corneal arcus. So here option A is correct. Then next question is, dash is the most common cause of fatty liver. So the most common cause of fatty liver is alcoholism. So here option D is correct. Next question is, dash is a condition of abnormal increase in body weight due to excessive fat deposition. So in the body, if excessive fat will deposit, that will lead to abnormally increase in the body weight, and that condition is known as obesity. So here option A will be the correct answer. The next question is, the DNA stands for. So DNA is deoxyribonucleic acid and RNA is ribose nucleic acid. So here option D is correct. Next question is, the hair protein is. So hair protein is a keratin. Keratin is a hair protein. So here option A is correct. Next question is, sucrose is a. So what is sucrose? It is a disaccharide. So here option D is correct. Next question is, the general test for carbohydrate is. So the general test for carbohydrate is Molish test. It is most important one. So it has been asked multiple times. So it is important. So option A is correct. Next question is, amino acids are linked together by a dash in protein molecules. So amino acids are linked together via peptide bond. So here option A will be the correct answer. Different amino acid A, B, C, these are linked via peptide bond. Next question is, in biochemistry PUFA stands for. So in biochemistry PUFA stands for polyunsaturated fatty acids. So here option A will be the correct answer. So the fatty acids can be saturated fatty acids or they can be unsaturated fatty acids. The saturated fatty acids are those in which there is no double bond in the structure, and unsaturated are those in which there are one or more double bonds in the structure. If there is only single double bond, then it is known as mono unsaturated fatty acids. If there are more than two or more than two uh, double bonds in the structure, then these are known as polyunsaturated fatty acids. Whole classification I have provided you in the form of table that you can check after the MCQs. Next question is, an acid contained in bacterial cell wall is. So the acid in the bacterial cell wall is muramic acid. Muramic acid. So here option A is correct. Next question is, the simplest amino acid is. So the simplest amino acid is glycine. So here option A will be the correct answer. So the glycine, the amino acids are also very important. There are two types of amino acid: essential and non-essential amino acids. So whole the classification I have provided you in the text form, we can discuss there, and also the classification of amino acid, uh, which is a amino acid on different areas like aspartic acid, glutamic acid, lysine. What are the symbols used for these amino acids? What is acidic amino acid, which is having aliphatic amino acid? So these all classification I have provided you in the form of table, so from there you can check. The next another question is, the an amino acid yielding acetyl coenzyme A during catabolism is. So this amino acid yielding acetyl catabolism during acetyl coenzyme A during catabolism is ketogenic. So here option A is correct. Next question is, the deficiency of protein alone is a symptom of. So this we have discussed in other subjects as well. It is the condition known as kwashiorkor protein alone, and protein plus carbohydrate deficiency is marasmus. So these are the two terms. Next question is, proteins gives blue to dash color with ninhydrin solution. So proteins they will give blue to violet color with ninhydrin solution. So it is a test for proteins. So here option C is correct. Next question is, Millen's test is given only by proteins having dash in their molecule. So Millen test is for tyrosine amino acid. So here option D is correct. Next question is, dash test is a specific reaction for the presence of arginine in proteins. So the for the presence of arginine, the test is Sakaguchi reaction. So here option D will be the correct answer. Next question is, alpha D glucose and beta D glucose both are. So these both are anomers. So here option B will be the correct answer. Next question is, compounds that have same structural formula what different spatial configuration are called. So those compounds which are having same structure formula but different spatial configuration, means their configuration will be different but the structural formula is same. So these are known as stereoisomers. So here option C will be the correct answer. Next question is, which of the following is epimeric pair? So epimers are glucose and mannose. These are the epimers. So here option D is correct. Next question is, one of the following is an enzyme required for glycolysis pathway. So the enzyme required for glycolysis pathway is pyruvate kinase. So here option B is correct. Next question is, primary structure of a protein is formed by. So the primary structure of protein is formed by the peptide bonds. So here option C is correct. Next question is, semi-essential amino acid is. So semi-essential amino acid is the histidine, so it is considered somewhere in some classification, it is considered in other form, but it is a semi-essential amino acid. So here option B will be the correct answer. And the essential and non-essential amino acid, this classification I have given you in the text so that you can read. Next question is non-protein amino acids are. So the non-protein amino acid is ornithine. So here option A will be the correct answer. Next question is the basic amino acid is. So among these amino acid like lysine, proline, leucine, tyrosine, the basic amino acid is lysine. So here option A will be the correct answer. And proline is the amino acid. It is the amino acid. Then next question is zymogen is. So zymogen is an inactivated enzyme. So here option A will be the correct answer. Next question is xanthoprotic test is positive for. So xanthoprotic test it is positive for aromatic amino acids. So here option C is correct. Next question is low plasma level of tryptophan and other neutral amino acids leads to the disorder known as. So if there are low levels of tryptophan uh, and other neutral amino acids, so then this condition is known as Hartnupp's disease. So here option C is correct. Next question is a test to evaluate the detoxifying function of liver is. So to evaluate the detoxifying function of liver, the test is hypuric acid test. So here option C is correct. Next question is for determination of amino acid sequence of a protein, dash is used. So in order to determine the amino acid sequence in a protein, the test used is Sanger's reagent test. So here option T is correct. Next question is holoenzyme is. So holoenzyme is a functional unit of enzyme which is made up of apoenzyme and coenzyme. So here option D, all of these is correct. Next question is enzymes which are produced in inactive form in the living cells are called. So those enzymes which are produced in inactive form in the living cells, those are known as proenzymes. So here option C is correct. Next question is in reversible non-competitive enzyme activity inhibition. So in case of reversible, it is a reversible non-competitive enzyme inhibition. The condition is concentration of active enzyme is decreased. So here option D will be the correct answer. Then the next question is the specificity of the enzyme is most dependent on. So the specificity of enzyme it is most dependent on the apoenzymes. So here option B is the correct answer. Next question is Zellweger syndrome occurs due to the Zellweger syndrome it occurs due to the presence of peroxisomes. So here option A will be the correct answer. Next question is the non-protein, organic and low molecular weight substance bound to an enzyme and essential for the activity of enzyme is known as. So this non-protein or organic low molecular weight substance it is the coenzyme. So here option B will be the correct answer. Next question is chemically Warford's reagent is. So the chemical composition of Warford reagents is copper acetate and glacial acetic acid. So here option A will be the correct answer. So the general test also I have mentioned in the form of table so that you can read as well. Next question is the oxidation of glucose to pyruvate and lactate is known as. So the oxidation of glucose to pyruvate and lactate. So this is the metabolic process known as glycolysis. So here option A is the correct answer. Next question is citric acid cycle is also known as. So the citric acid cycle it is also known as Krebs cycle. So here option C is correct or TCA cycle. Next question is Embden-Meyerhof pathway is also known as. So Embden-Meyerhof pathway it is the another name for glycolysis. So here option D is correct. Next question is one of the following is oxidative pathway of glucose. So the oxidative pathway of glucose is HMP shunt pathway, that is hexose monophosphate shunt pathway. So option B is correct. Next question is the inhibition of glycolysis by oxygen is known as. So the inhibition of glycolysis by oxygen, it is known as Pasteur's effect. So here option B is correct. Next question is the enzyme of tricarboxylic acid cycle are located in. So the enzymes of TCA cycle, these are located in the mitochondrial matrix. So here option B is correct. Next question is Von Guy's disease occurs because of. So Von Guy's disease, it occurs due to the accumulation of glycogen in hepatocytes in the renal cells. So here option C will be the correct answer. Next question is in hexose monophosphate shunt, glucose 6 phosphate is converted into 6 phosphogluconolactone by. So in HMP shunt pathway, there is a step in which glucose 6 phosphate is converted to 6 phosphogluconolactone. It is preceded by the glucose 6 phosphate dehydrogenase enzyme. So here option A will be the correct answer. So for these type of questions, you need to read all the cycles like glycolysis, glycogenolysis, HMP shunt pathway, TCA cycle, so uric acid cycle. So these all cycle you need to learn. So that you, for these cycles you can refer to any biochemistry book, for example biochemistry by Satyanarayan. In that book you will be finding all these cycles. Then the next question is Gaucher's disease occurs due to the deficiency of. So Gaucher disease, it occurs due to the deficiency of beta glucosidase. So here option D is correct. Next question is the sugar present in RNA is. So the sugar in RNA is ribose sugar. So here option A is correct. Next question is urea cycle is known as. So urea cycle is also known as Krebs-Henseleit cycle. So here option B is correct. Next question is Krabbe's disease occurs due to the defect in. So Krabbe's disease, it occurs due to the defect in beta galactosidase. So here option B is correct. Next question is which of the following test is specific for the ketone bodies? So for ketone bodies, the specific test is Rothra's test. So here option A is correct. Next question is which of the following tests are positive for bile salts? So for bile salts, Hayes test and Patton Cofer's test, these both tests are positive. So here option C will be the correct answer. Next question is which of the following test is specific for ketohexosis? So for ketohexosis, the test is Selivanov test. So here option D is correct. Next question is which one is described as vitamin in search of disease? So the vitamin in search of disease is known as vitamin H. So here option D is correct. So these are the few of the questions, around 100 MCQ questions we have discussed. So here I have provided you few tables from the biochemistry book which are also important. So here first table is regarding the glycogen storage disease. If you have that biochemistry book by Satyanarayan, then you can refer to that book. You will get all these things. Rest I have provided you many things in this particular PDF, so you can refer to this as well. So here the first disease is, these all are the glycogen storage disease. Von Guy's disease, it is very important. It is the type 1 glycogenosis disease. Enzyme defect is glucose 6 phosphate, and organs involved are liver, kidney, and intestine. So these characteristics you can read. Then there is Pom's disease, which is due to the defect in the lysosomal alpha 1 for glucosidase. Uh, and so in these, the von Guy's disease is very important. It has been asked previously. Then Cory's disease is also important. You can read this as well. The amylo alpha 1,6 glucosidase debranching enzyme is involved, and it is also known as Fauve's disease. Then there is Anderson disease, which is known as amylopectinosis, which is due to the defect in glucosyl 4,6 transferase. Then the disease is McArdle's disease, which is a type 5 glycogenosis, which is due to the defect in muscle glycogen and phosphorylase. Then the next disease is Hertz disease, which is due to liver glycogen and phosphorylase only. And then the Tourette's disease, which is due to the defect in phosphofructokinase. So these all things you can remember. The naming things are always very very important. Then the next table I have added you regarding the classification of fatty acids, which are saturated fatty acids in which there is no double bond present. So sometimes the question asks regarding the number of carbon atoms. So here the first term, that is given 2, 3, 4, 5, 6, 8, 10, 12. These are the number of carbons present in that specific fatty acid. So arachidic acid has been asked previously, like how many carbon atoms are present in arachidic acid and palmitic acid. These two have been asked previously in the question papers. So in palmitic there are 16 carbon atoms, in arachidic acid there are 20 carbon atoms. And in saturated fatty acid there are no double bonds, that's why all the numbers are 0. Then moving to the next, that is the unsaturated fatty acids. So these are the fatty acids in which there are one or more double bonds. Okay, so here the name is given, and these the first digit represents the number of carbon atoms. Here also arachidonic acid is given, has been asked previously. So here are 20 carbon atoms present, and there are multiple double bonds. So at which position the double bond is there, so these positions are mentioned here. So here you can see, like in these are the structures, 1 2 3 4 double bonds are there, and you can check the position 4 5 8 11 14. So these are the different position where the double bonds is present. So these you can remember, like which of the following are fatty acid, saturated fatty acid, which are unsaturated fatty acid. So these you can see. Then moving to the next thing, that is the examples of sphingolipidosis, the lipid storage diseases. These are also very very important. So few question I have added. So in this I'll tell you, like which question has been asked previously. So name and pick disease has been asked, Farber's disease, Gaucher disease, Graves and Fabry's. So these are the different diseases which has been asked. Mostly in this, the enzyme is mainly asked, like due to which enzyme or which defective enzyme this particular disease is asked. So name and pick disease, it is asked multiple times, 2 to 3 times. And in the SSSB exams, these Farber's disease, Gaucher disease, Graves disease, so these are asked in those different exams. So these you can read, like the enzymes and the name of the disease. Then the next one is the classification of amino acid, the amino acids with aliphatic R group. So first is 3-letter abbreviation, and then proceeding to that there is a symbol that is used. So these all things you can remember. Some symbols which are different from the first letter of the amino acid, so those you need to remember. So here the non-aromatic amino acid with hydroxyl R group, serine and threonine, sulfur containing compounds. These are very very important. Many a times this has been asked, like which of the following are sulfur containing amino acids. So cysteine and methionine, these are the sulfur containing amino acids. Then there can be asked, like which of these is amide containing amino acid. So there are four: aspartic acid, asparginase, glutamic acid and glutamine. These are the amide containing, and the symbol used for this is different. Like for aspartic acid it is D, for asparaginase it is N, glutamic acid it is E, and glutamine it is Q. So these are the different, so these you need to remember because these are having the different symbols. Then the next is basic amino acids: arginine, lysine, and histidine. So one question that I have included, like which of the following is basic amino acid? So the answer was lysine. So in that way, the question can be transformed in different ways. Then here also the symbols are different, like R, K, for histidine it is H only. So these are different things you can remember. Then there are amino acids with aromatic rings. This is very important. Like the aromatic amino acids are the examples of aromatic amino acid. These questions have been asked. So phenylalanine, tyrosine, tryptophan. So these are the different things. For phenylalanine, F is the symbol, tyrosine Y is the symbol, tryptophan W is the symbol. So this question has been asked recently in one of the exam for the symbol of tryptophan, so which is W. So this you can remember. Then the next is the amino acid. So amino acid, I have told you, like it is the proline, which is also known as amino acids. Then there are mainly two types of amino acid as per the nutritional requirements. One are essential amino acid and the other is non-essential amino acids. Essential amino acids are those which are not synthesized by the body and they are needed in the diet. So here the example is this histidine is the essential amino acid, but sometimes it can be transformed in the body as well. So it is a semi-essential amino acid. It can be considered as semi-essential amino acid. Then in the non-essential amino acids, these are the different non-essential amino acids which are not required in the diet, but they are synthesized in the body itself. For example, glycine, alanine, serine, cysteine, tyrosine, proline, aspartic acid, glutamic acid. These all are the non-essential amino acids. Then we can move to next part, which is the definitions of different metabolic processes of carbohydrates, which are very very important. So for glycolysis, it is the oxidation of glucose to pyruvate and lactate. Means basically transformation of glucose to pyruvate and lactate. Sometimes the term degradation is used, like the degradation of glucose to pyruvate and lactate. So this can be asked. Then the citric acid cycle, which is also known Krebs cycle or TCA, that is tricarboxylic acid cycle. And in this, the oxidation of acetyl coenzyme A to carbon dioxide happens. So Krebs cycle is the final common oxidative pathway for carbohydrates, fat, amino acids through acetyl coenzyme A. Then gluconeogenesis, the synthesis of glucose from non-carbohydrate precursor. This is also very important. Directly question is asked, like in which process the glucose is synthesized from non-carbohydrate source. So that is the gluconeogenesis. The next is glycogenesis, glycogen. So glycogen means formation of glycogen from glucose. Then glycogenolysis. Lysis means breakdown. So breakdown of glycogen to glucose. So these two terms are slight different, slight different in the naming and in the working as well. So basically from the name itself you will be able to remember these terms. The next is hexose monophosphate shunt pathway, which is also known as pentose phosphate pathway or direct oxidative pathway. So this is alternative pathway to glycolysis and TCA cycle for the oxidation of glucose, which is directly to carbon dioxide and water. Then there is a uronic acid pathway in which the glucose is converted to glucuronic acid, pentosis, and in some animals to ascorbic acid as well. And this pathway is also an alternative oxidative pathway for glucose as well. Then there are galactose metabolism, fructose metabolism, amino sugar and mucopolysaccharide metabolism. These are the different mechanism or different pathways for carbohydrate metabolism. So these you can read. All the text has been provided to you. Then next I have added several tests for the carbohydrate, lipids, and amino acids. So these you can read. So from here there is a chemical constant like carbohydrate, the different test, Molish test, which is for general sugar, means all the monosaccharides, or carbohydrate, the general test is Molish test. Then for reducing sugars, the test is Felling's test or Benedict test. You can also remember the color. Sometimes color can be asked. Then for monosaccharides, Warford test is there. Then for pentosis, there is Weil-Sorsinol test. For hexose, there can be Selvinov test. For starch, there is iodine test. For gums, there is Felling's test and Benedict test. For mucilage, ruthenium red test is there. Then for proteins, we have Burette test, which is for which is a general test for proteins. Millens test is there. Then there is xanthoprotic test, which is for tyrosine and tryptophan, phenylalanine containing proteins, basically for aromatic amino acids. Then there is the precipitation test for proteins as well. Then for amino acids, there is a test, nin hydrogen test, which is a general test, and a specific test is Millen's reagent test, that is for tyrosine. And for cysteine, and there is a test in which the sodium hydroxide is used in combination with 10% lead acetate. If the cysteine is present, then the black precipitate of lead sulfide will form. So these are the different tests. At least you have to remember the names of these tests. Then moving to next topic, which is the brief chemistry and the role of vitamins, enzymes, coenzymes, and minerals. So let's start like vitamins, enzymes. These we have discussed in other subjects as well. So we will have a quick revision of these few of the MCQ questions that you can remember. So the first question is, which of the following vitamin is also called as water soluble vitamin? So the fat soluble is A, D, E, K, and water soluble is vitamin B and C. So here option C will be the correct answer. Next question is, which of the following vitamins are known as fat soluble vitamin? The fat soluble vitamin is vitamin E. So option B is correct. Next question is, which of the following is the most essential nutrient for a woman during her initial stages of pregnancy to prevent birth defects? So this most essential nutrient is folic acid and also the calcium. So here option B will be the correct answer. Next question is, which of the following vitamin helps in blood clotting? So for blood clotting, the vitamin essential is the vitamin K. So here option D is correct. It is involved in four clotting factors: clotting factor number 2, clotting factor number 7, clotting factor number 9, and clotting factor number 10. These four clotting factors, the synthesis is done by the vitamin K. Next question is, which of the following is a leading cause of blindness in children worldwide? So leading cause of blindness in children is vitamin A deficiency. So here option D will be the correct answer. Then the next question is, which of the following vitamin deficiency causes very? So very is caused by the vitamin B1 deficiency. So here option A is correct. Next question is, which of the following nutrient deficiency causes megaloblastic anemia? Megaloblastic anemia is caused due to folic acid deficiency, so option A is correct. Next question is, which of the following diseases caused by the deficiency of niacin? So for niacin, pellagra is caused, so option C is correct. Next question is, which of the following vitamins serves as hormone precursor? So the hormone precursor is vitamin D. So it is also known as steroidal vitamin. So here option C is correct. Next question is, which of the following is a component of the coenzyme A? So the component of coenzyme A is pantothenic acid. So here option D is correct. Next question is, which of the following vitamins is also known as covalamin? So covalamin or cyanocobalamin, this is vitamin B12. So option D is correct. Next question is, fat soluble vitamins are stored in. So the fat soluble vitamins, these are stored in the liver. So option D is correct. Next question is, which of the following vitamin is not excreted out of the body? So the vitamin which is not excreted out of the body is vitamin E. So here option B is correct. It is also known as tocopherol. Tocopherol, and the most active form is alpha tocopherol. Next question is, thiamine is a scientific name of. So thiamine, it is a scientific name of vitamin B1. So option C is correct. Next question is, vitamin A is also known as, so vitamin A is also known as retinol. So here option B will be the correct answer. Next question is, the nutritional content of dash is defined in terms of chemical activity of alpha tocopherol. So alpha tocopherol is a active form of vitamin E, and it is The correct option will be option number B, vitamin E. So option B is correct. Next question is, vitamin K is also known as, so the another name for vitamin K is phyloquinone. So here option C is correct. So there are three forms of vitamin K: vitamin K1, K2, K3, and you have to remember the names. Vitamin K is phyloquinone, vitamin K is menaquinone, and vitamin K3 is menadione. Next question is, riboflavin is, so riboflavin is vitamin B2. So here option A is correct. Next question is, the dash molecule is composed of three separate parts called pyridine, paraminobenzoic acid, and glutamic acid. So these three different parts are in folic acid. So here option A is correct. Next question is, dash is a hexose derivative and it exists in two forms, L-ascorbic acid and T-ascorbic acid. So this one is ascorbic acid only. From the question itself, the answer is clear. So option B is correct. Next question is, dash is also known as folacin or vitamin B9. So vitamin B9 is folic acid. The folacin term is used, so it is folic acid only. So option C is correct. Next question is, dash is a B complex vitamin also known as vitamin B7 and is water soluble. So vitamin B7 is biotin. So here option D is correct. Next question is, ascorbic acid is also known as, so the ascorbic acid is vitamin C. So option B is correct. Next question is, small organic molecules transporting the chemical groups from one enzyme to another are known as. So the small organic compounds which are used for transporting the chemical groups from one enzyme to another, these are the coenzymes. So here option D is the correct answer. Next question is, dash is a strong reducing agent. So ascorbic acid is a strong reducing agent. So here option A will be the correct answer. Next question is, according to International Union of Biochemistry, IUV, enzymes has been classified into dash major classes. So according to IUV, the enzymes are classified into six major classification. So here option B will be the correct answer. What are the six these main classes? So first one is oxidoreductases. I have added the table for this classification as well, so you can read the table also. The second one is transferases. Then the third one is hydrolases hydrolases. Then the fourth one is lyases. Fifth one is isomerases, and the sixth one is ligases. So these are the different uh, classes, and the examples and what type of reactions are catalyzed, those you can read in the table itself. Then the next question is, dash are the catalytically active enzyme made up of coenzyme or metal ions, that is the non-protein part, and apoenzyme or apoprotein, which is the protein part. So with this question you will be able to uh, remember the whole question, like each and every part, like the coenzyme or metal ion, it is the non-protein part, apoenzyme is the protein part. So sometimes the question can be asked like, which of the following is the protein part? So that is the apoprotein, which is the non-protein part, that is the coenzyme. So these are the different you can say these are the holoenzymes, which are catalytically active enzymes, and they are made up of these both coenzyme and apoenzyme. So option D is correct. Next question is, the study of enzyme catalyzed reaction is known as, so the study of enzyme catalyzed reaction, it is known as enzyme kinetics. So here option D is correct. Next question is, the enzyme catalyzed reaction rate and dash are directly proportional to each other. So the enzyme catalyzed reaction rate, okay, simply reaction rate and concentration, these are the directly proportional to each other. So here option B will be the correct answer. Next question is the optimum temperature for most of the enzymes is between. So the optimum temperature for most of the enzyme is between 40 to 45 degrees Celsius. So here option B is the correct answer. Next question is optimum activity of several enzymes can be seen between the pH ranging from. So the optical activity of several enzymes that can be seen between the pH ranging from 5 to 9. So here option D is correct. Next question is dash is used for treating leukemia. So for treating leukemia, asparaginase enzyme is used. So option B is correct. Next question is urokinase and dash drugs are used for treating blood clotting. So urokinase and nattokinase, these drugs are used for treating the blood clotting. So here option C is correct. Next question is dash enzyme is used as antiviral agent. So the antiviral agent is ribonuclease. So here option B is correct. Next question is collagenase is used in the treatment of. So collagenase it is used in the treatment of skin ulcers. So here option A will be the correct answer. Next question is serum glutamate oxaloacetate transaminase is used in the diagnosis of. So serum glutamate oxaloacetate transaminase SGOT. So it is used in the diagnosis of heart attack. So here option D is the correct answer. Next question is dash is used in the diagnosis of infective hepatitis, leukemia, muscular dystrophy, and myocardial infarction. So these all are the lactate dehydrogenase. So lactate dehydrogenase it is used in these different uh, diseases diagnosis conditions. So here option A is correct. Next question is dash is used as a good tumor marker. So tumor marker is acid phosphate. So here option D is correct. Then the next question is dash are the metal ions or coenzymes which bound covalently, evenly and tightly to the enzyme proteins. So these are the prosthetic groups which are the metal ions or coenzymes that bound covalently to the enzyme proteins. Next question is dash enzymes is used for the diagnosis of muscular dystrophy. So the enzyme which is used for the diagnosis of muscular dystrophy is aldolase. So here option D will be the correct answer. So these are the few of the MCQ questions that are in the vitamins and enzymes classes. And the other important things, like I have only included few of the MCQ questions, rest I have added you in the this text part in the form of table. So here this is the IUV classification of enzymes. 6 classes are there. You can remember the number of class, like sometimes the question has been asked previously, like which is the fourth class of enzymes based on the IUV classification. So that is the lyase. So these type of question has been asked. So the order you need to remember, what type of reaction catalyzed in these particular things, that you can remember, and the examples you can remember. So here are 6 classes. You can remember the classes, examples, and also the reaction conditions. Then the next table I have added is for the classification of vitamins. So from this you can remember the what are fat soluble, what are water soluble, what are the names of like vitamin B1, B2, B3, B6, B7, B5. What are the names, which vitamin is known as B3, which vitamin is known as B7. So these names you can remember uh, from this table. Then there are several examples of enzymes and their respective substrate and competitive inhibitors. So these you can remember. Sometimes inhibitors can be asked or substrate can be asked. Mostly the drugs are used in pharmacology aspect as well. So you can remember here as well for revision part. So these things you can remember. Then the coenzymes of B complex vitamin, these are very important. Sometimes the question asked regarding the which of the following is the coenzyme of vitamin pantothenic acid, vitamin B5, So these kind of things can be asked. Like coenzyme A is the coenzyme of pantothenic acid. The PLP is the is for pyridoxine. FMN has been asked. FAD has been asked. TPP has been asked. NAD. So these all things you can remember from here, from this table. Then the next table is regarding the selected list of applications of enzymes. So there are different enzymes and the application of these enzymes, like papain, it is anti-inflammatory, streptokinase, urokinase, these are used to remove blood clot, asparaginase in cancer therapy. This is important, asparaginase is important. Uh, and then analytical for basically for the diagnosis part, for estimation, then in genetic engineering, industrial application. These main you can remember. Like for asparaginase, I have told you this question has been asked. Rest you can simply have a reading on these things 1 or 2 times, so that it will have a memory of you to solve the MCQs in this particular way. The next, I have added you the important enzymes in the diagnosis of disease. So as got this question I have added, like it is for heart attacks or heart conditions. Then SGPT, it is for liver diseases. Amylase, it is for acute pancreatitis. Alkaline phosphate is for rickets, obstructive jaundice. Acid phosphatase is cancer of prostate gland. Lactate dehydrogenase, it is for heart attack and liver disease. Creatinine phosphokinase, this is for myocardial infections. Aldolases, these are for muscular dystrophy. 5-nucleotidase, these are for hepatitis. And gamma glutamyl transpeptidase, this is for alcoholism. So SGPT, SGOT, these are very important. Alkaline phosphate is important. Lactate dehydrogenase is important, and aldolase. These are the few of the examples which are very important in this table. So these are the few things regarding the vitamins enzymes part. Now we can move to the last topic, that is the therapeutics, in which we will be having a discussion on the pathology of blood and urine, lymphocytes, which is a part of blood, erythrocytes, RBCs, lymphocytes, WBCs, then abnormal constituents of urine and their significance in disease. The different topics will be covered in these MCQs. So let's start with the first question. The question is, a branch of medicinal science which diagnose disease on the basis of laboratory analysis of body fluids and tissues is known as. So this branch is basically known as pathology. So here option D is correct. Next question is, dash is a branch of medical science concerned with the study of blood, blood forming tissues and blood disorder. So this branch of science is hematology. So here option B is correct. Next question is, dash is formed in the liver and is the most abundant plasma protein. So which is the most formed one? Formed which protein is formed in the liver and most abundant plasma protein? So this most abundant plasma protein is albumin. So here option A is correct. Next question is, dash helps in the maintenance of osmotic pressure in the blood vessels. So in the blood vessels, fibrinogen is responsible for the maintenance of osmotic pressure in blood vessels. Option B is correct. Next question is, dash is the key component of blood clotting and solidifies from liquid to fibers, which controls bleeding. So this one is fibrinogen. So here option D will be the correct answer. Next question is, dash maintains osmotic pressure while retaining fluid in vascular compartments. So globulin is the protein which maintains the osmotic pressure by retaining fluid in the vascular compartments. So here option A will be the correct answer. Next question is, thrombocytopenia is caused due to the disorder of. So thrombocytopenia, it is caused due to the disorder of platelets. So here option B is correct. Next question is anemia is caused due to the disorder of. So anemia is caused due to the disorder of erythrocytes. So here option A is correct. Next question is leukemia is caused due to the disorder of. So leukemia it is caused due to the disorder of leukocytes. So option D is correct. Next question is hemophilia is caused due to the disorder of. So hemophilia it is due to the disorder of clotting factors basically. So when I have discussed with you regarding the 13 clotting factors, in that I have discussed with you like the clotting factor 8, clotting factor 9, clotting factor 11 and clotting factor 12. So 8 is known as anti-hemophilic factor number A, then it is anti-hemophilic factor B, it is anti-hemophilic factor C, it is anti-hemophilic factor D. So in that way these all are used, these all are the different factors. I have included the question as well. There you will be having the more clarity like what type of question can be asked. Next question is disorders of the erythrocytes cause. So the disorders of erythrocytes they cause polycythemia. So here option D is correct. So polycythemia is increase in the number of RBC. Increase in the number of RBC is polycythemia. So this term is very very important. Next question is dash anemia occurs due to decreased marrow function and aplastic anemia occurs due to destruction of bone marrow. So the anemia which occurs due to decreased marrow function is hypoplastic anemia. So here option B is correct. Next question is primary polycythemia is a consequence of pathological condition is known as. So primary polycythemia it is due to the pathological condition, consequence in pathological condition known as polycythemia vera. So option D is correct. Next question is dash is caused by chronic hypoxia as a result of long standing lung disease or cardiac failure. So this condition is known as pathological polycythemia. So option D is correct. Next question is dash occurs if a person migrates from sea level to high altitude level. So these are the physiological polycythemia which occurs if a person migrates, if there is a migration from sea level to high altitude level. So then the option will be option number C, physiological polycythemia. Next question is dash is a cancerous condition characterized by multiplication of abnormal WBCs in an uncontrolled manner. In an uncontrolled manner. So this condition is known as leukemia. So here option C is the correct answer. It is related to white blood cell WBCs. Next question is dash is required for the synthesis of clotting factor 2, 7, 9, 10. So this is vitamin K. So option A is correct. Next question is dash is the most common form caused due to deficiency of clotting factor A. Clotting factor 8. So due to the deficiency of clotting factor A, hemophilia A is caused. So option B is correct. Next question is dash results due to the deficiency of clotting factor 9. For clotting factor 9, hemophilia B is the condition. Then next question is a mild form resulting due to deficiency of clotting factor 9. So due to sorry not 9, clotting factor 11. So for clotting factor 11 the condition is hemophilia C. So option A is correct. And for clotting factor 12 the condition is hemophilia D. Next question is normal mature erythrocytes are disc shaped. Why concave cells which do not have? So the mature RBCs they do not have the nucleus. So here option A will be the correct answer. This we have discussed in the human anatomy part as well. So this question is also important. Next question is a common term used for an increased variation in cell size of erythrocytes is known as. So increased variation of cell size, increased variation in cell size of erythrocyte, it is known as anisocytosis, and increased number of RBCs is known as polycythemia. Next question is a nuclear maturation defect resulting due to deficiency of either vitamin B12 or folate is known as. So this condition is known as true macrocytes. So which is a nuclear maturation defect which is due to the deficiency of vitamin B12 or folic acid vitamin B9. Next question is in dash synthesis of hemoglobin decreases due to the deficiency, iron deficiency, impaired globulin synthesis and mitochondrial abnormality. So this condition is microcytosis. So in this the hemoglobin synthesis decreases. Then the next question is a term dash is used to denote mature erythrocytes that have the shape other than the normal round Y concave appearance on stained blood smear or variation. So this condition is poikilokyrosis. So it is used to denote the mature erythrocytes which is having a different shape like shape other than normal round or Y concave. So these are the poikilocyrosis. Next question is dash have multiple thorny spike-like projections which are irregularly distributed around the cellular membrane and may vary in size. So these conditions are acanthocytes. So here option B is correct. They have multiple thorny spike-like projections and they are irregularly distributed around the cellular membrane. Next question is dash have one or more vacuoles which look like a blister on the skin and have thinned area at the periphery of the cell membrane. So these all are the blister cells. So here option D is correct. Next question is dash have one or more spiny projections of cellular membrane. So one or more spiny projections of cellular membrane, these are the Wurth cells which are having these type of structures. So option A is correct. Next question is dash results from the production of abnormal cells via the bone marrow or from damage to normal cells. So this condition is poikilocytes. So poikilocytes, these results from the production of abnormal cells. So option D is correct. Next question is dash have short scalloped or spike-like projection which are regularly distributed around the membrane. So these are regularly distributed. So these are the echinocytes. So option B is correct. Next question is dash have road, cigar or sausage like shape and appear narrower and more elongated than megalocytes. So these all are the characteristics of elliptocytes. So here option A is correct. Next question is dash are found when there is disturbance in the synthesis of DNA due to vitamin B12 or folic acid deficiency. So this condition is known as oval macrocytes. So option D is correct. The next question is dash are fragments of red blood cells and are formed either by fragmentation of abnormal cells. So these are schistocytes which are the fragments of RBCs and they are formed either by fragmentation of abnormal cells. Next question is dash are formed in sickle cell anemia. In sickle cell anemia, drepanocytes are formed. So here option A is correct. Next question is dash are spherical or nearly spherical in shape rather than Y concave disc shape. So these are the spherocytes. So option B is correct. Next question is dash shows a reduced staining of RBCs due to an increase in central pallor. So this is the condition is hypochromia. It shows a reduced staining of RBC. So option A is correct. Next question is red blood cells have iron containing oxygen transporting metalloprotein known as. So in RBC is the oxygen transporting protein is hemoglobin. So option B is correct. Next question is, dash term is used for cells that are more intensely stained than normal or to indicate the hyperchromic cell condition. So the term used for this condition is hyperchromia. So here option C is correct. Next question is, presence of abnormal concentration of albumin and globulin in the urine is known as. So if there is abnormal concentration of albumin and globulin protein in the urine, that condition is known as proteinuria. So here option A is correct. Next question is, presence of glucose in urine is known as. So the presence of glucose in urine, so that condition is known as glycosuria. So option D is correct. Next question is, presence of fructose sugar in urine due to disturbed fructose metabolism is known as. So presence of fructose sugar. So it is a condition known as fructoseuria. So option B is correct. Next question is, a condition in which wild salts are excreted in urine is known as. So a condition in which wild salts are excreted in urine, this condition is known as bilirubinate bilirubinuria. So option D is correct. Then next question is, presence of RBCs in urine. Presence of RBCs or blood in urine, it is known as a condition known as hematuria. So option B is correct. Next question is, during excessive hemolysis, part of the bile pigment formed by the breakdown of hemoglobin is excreted out by urine, which is known as. So when there is excessive hemolysis, then a part of bile pigment it formed by the breakdown of hemoglobin, means hemoglobin will break down and bile pigment will be formed, and this part will be excreted in urine, and this condition is known as urobilinogen. So here option A is correct. Next question is, consumption of food rich in pentose sugars like grapes, cherries, and plums causes. So this condition can cause pentoseuria. So here option B will be the correct answer. So with this question, we are done with this particular unit number 9. We have covered all the topics, all the important things I have mentioned you in the form of MCQs. The rest I have provided you in the form of tables from books, several books, so that you can read on your own and have a better understanding on the concept, on the things. So yeah, so that's all for this video for the unit number 10. I will be upload unit number 10 today only. If it is not completed by today, then mostly it will be done by the tomorrow morning or tomorrow evening. So yeah, that's all for this video. Thank you very much.